Welcome to this new session on RDA and similarity measures as well as non-metric multidimensional scaling. So the learning targets for today are to understand the basics of RDA, to gain some knowledge on the calculation of commonly used association measures, so similarity measures, to understand their suitability for ecological data, so the suitability of different association measures, and to understand the mathematical background and how to conduct a non-metric multidimensional scaling, the acronym of which is NMDS. So here you have the learning targets and the associated study questions. On this slide as well, the study questions related to the mathematical background and how to conduct an NMDS. Let's begin by recapturing what we have done in previous parts of the course. In simple ordination, we dealt, and this is shown in the figure A on the top left, we have a matrix of variables and we have objects. On our examples, these were measurements of environmental variables and in different sites, and we use these to run a principal component analysis. A different form of analysis would be a correspondence analysis that assumes a unimodal gradient across the environmental space for a species and is more useful to ordinate species distances. Under B, you see the example of multiple regression analysis. So you have the variable y, which is a vector here. For example, we had species richness in our course measured in different sites, and you have several variables that can be used to explain these responses. So for example, you fit environmental variables, that's shown in the model. You use the environmental variables to fit the responses where you expect in the linear regression model that the deviations from the fitted values are normally distributed. So we now turn to the case of ordination of a matrix of y. That means we have several response variables compared to the case of multiple regression where we had just one response variable. So we have a vector of response variables. That could be a matrix of species responses, species data, or other response variables. And in the ecological context, most often you have species data and variables that may influence the communities that are represented by the matrix y. The variables can be any environmental variable, a stressor variable, and so on. So the aim of redundancy analysis is to display and explain variation in the set of response variables constrained by a second set of predictor variables. So this is a constrained ordination method that term that we have discussed before. It's an ordination where only the data are extracted that are explained by a second data set. So this technique links multivariate multiple regression analysis and PCA, and you will in the next slide see why this is the case. An example where this technique has been applied is in a study where we analyzed the occurrence and the densities of different fungal species along gradients of environmental variables, and we ask which variables do best explain the variation in the fungal communities sampled along a gradient of fungicide toxicity. And indeed it was in the end the fungicide toxicity that had that was the only variable that exhibited statistically significant explanatory power. And in the plot on the bottom of this figure, you can see how the different fungal organisms responded, the species, the taxa responded to the toxicity in the sites. So this reads similarly to what we have discussed in the PCA before. So species that are closer together share a similar response, and those that are further apart are occurring in different sites. So we see generally in this graph that some species that are like C. stellata are rather occurring when you have a higher toxicity, whereas Limnophila aquatica on the right side is not occurring when you have high toxicity. So remember how we conducted the multiple linear regression analysis in matrix form. It's just copy and paste from our slides that we discussed in the context of linear regression analysis. So we have the response vector of fitted, the response vector, and we have the obtain in multiple linear regression analysis a fitted response vector, that's the fitted y's. And this can be expressed in different terms. You see that on the slide, and the bottom of the slide you see the reformulation for the case of multivariate multiple regression analysis, where we have several vectors of y. So that's, loosely speaking, this is basically just a multiple regression where we explain each of the response vectors in the, for example, ecological community, all species that have been observed, we express them in terms of the linear, in a linear combination of environmental variables. This also means that the assumptions of the multiple linear regression model must be fulfilled, must be met to be meaningful, to the analysis to be meaningful. So how is it done? We don't go into complete technical details on this issue here, because many of the technical background has been discussed already in the context of PCA. And just to give you an idea where multivariate multiple regression and PCA are linked, is that the RDA uses the variance covariance matrix of the fitted Y matrix. So we first do the multivariate multiple regression, and then we use this fitted matrix to estimate the variance covariance matrix. Since we usually don't know this, we use the sample variance covariance matrix and estimate this from the data. And when we look now, that this looks similar to the univariate case, should be familiar to you. And this is then used, this sample variance covariance matrix is then used in the PCA, and you should see that we encounter again our eigenvalue problem with the eigenvectors. So we basically run a PCA just using a variance covariance matrix that has been obtained by multivariate multiple regression analysis. So the eigenvectors in the end then are linear combinations of our explanatory variables. Looking on this in a more formalized way, this figure may seem a bit complicated at first. So we go through this figure from top to bottom. In the first the top left, you have the data table. That's the centered variables. That's our response variables, for example, species. And we have beside that the explanatory variables X centered as well. And what we do first is we regress each variable Y on the table X and compute the fitted and residuals. So that is basically what we have already mentioned a couple of times. That's our multivariate multiple regression analysis. Then we take this and run a PCA on this and obtain the matrix of eigenvectors. And with this we obtain the ordination in the space of variables if we use the eigenvectors in conjunction with the fitted values from the multiple regression. So we obtain the fitted side scores, and on the other hand we can use the remaining non-explained variance which is left from the multiple regression analysis, and to run a PCA on this, and then we obtain the ordination in the space of residuals. So that's what we saw on the example case, that if you have just one explanatory variable, then you have just one RDA axis. So the RDA axis number always is the same as the number of explanatory variables. And the second axis in this case was a principal component axis based on the residuals, on the non-explained variance. What is the result of an RDA? First of all, compared to the PCA, where we can have a plot of environmental variables and sites, we can have a plot which was a B plot in the context of PCA. In the context of RDA, we may obtain a tree plot. So we can have a plot that is shown on this slide that gives you the relationship between species, sites, and environmental variables. And you read this plot in the same way as we have discussed previously for the PCA. So the distance between objects indicates how similar the sites are. The correlation can be estimated between species when you compare the angle between the arrows, and the same holds for environmental variables and so on. So the interpretation is pretty similar. So in this case we would interpret that aluminium is one of the driving force on the first axis of the communities, and there are some species that are in the same direction as aluminium along the first axis. They rather occur at high aluminium sites, while other species occur in the opposite side. For example, VAC point VIT would be most sensitive to aluminium if the correlations that we have found are causal. In the same way, we would interpret that, for example, you have the lowest aluminium concentrations in site 21 and the highest perhaps in site 5. There are other issues regarding the scaling, so whether the distances can be expressed in reliable form in the figure, or whether the correlations are more and can be reliably interpreted. So different types of scaling that is discussed in the context of the implementation in R. You obtain eigenvalues and variance partitioning. So you obtain the constraint variance, the variance that is explained by the environmental variables, and the unconstrained variables, so the residuals that are left after fitting the environmental variables. You obtain site scores and species scores and the B-plot scores for variables. But that's rather technical details that we will have a look at in the context of the R demonstration. So similar issues that have been discussed in the context of PCA need to be tackled in the context of RDA. First of all, you have the question as well, how many RDA axes do we need? And here we can actually use a hypothesis test based procedure that's based on permutations, so it's not test based on a test statistic. And this has been recommended as method in a paper like Legendre et al. Methods in Ecology and Evolution 2011. So this is what we have used as well in the R script. And another question is, how many environmental variables are really needed and how important are they? On the one hand, to tackle how many environmental variables are needed, this is the same question that we have faced in the context of multiple linear regression analysis. If you remember what the method that we used there, one of the goodness of fit metrics we discussed was the adjusted R squared, and that has been implemented for model building in R as well. For the RDA, so we can use manual and automatic model building with the adjusted R square. So remember that you could, for example, build a couple of competing models, ideally based on scientific hypothesis, and compare their fit to the data. Or on the other hand, you can run automatic model building. We have discussed extensively the shortcomings of just letting the computer do the model building with automatic model building, and in the context of multiple linear regression analysis. On the other hand, you have the possibility to interpret, when you have fitted a best fit model, you can interpret how important the variables are based on the variance they explain on their so-called methods of variance partitioning, that tell you how much in the explained variance, how much an individual variable explains in terms of variance. There are a couple of assumptions of the RDA, and these are similar to what we have heard before in the context of multiple linear regression analysis. On the one hand, you assume the independence of observations, so of the sites. Then you assume that you have a linear relationship between explanatory and response variables that needs to be checked, and we discussed briefly how this is done in the context of the R script and on the next slide. You assume that there is no multicollinearity between explanatory variables, so in the data preparation you actually check a couple of things that we have checked in multiple linear regression analysis as well. For example, we use the variance inflation factors to check for and plots to check for multicollinearity between variables, and we look at the distribution of individual variables to evaluate whether they need transformation. And another issue that is sometimes done, or another data preparation step that is sometimes done, is that rare species are removed before RDA. For example, if we try to establish a linear gradient or linear response of species to environmental variables, and you have a species that just occurs at 1 or 2 slides of 20-30 sites, or let's just say at 1 or 2%, a few percent of sites, then you cannot establish a linear relationship with an environmental variable. So sometimes these are removed from the data set before RDA to strengthen the relationship between predictors and the species on the first few axes. There are a few extensions of RDA. For example, RDA can be used for multivariate ANOVA. This is discussed extensively in Bray-Curtis et al. We will briefly look into this in the context of multivariate hypothesis testing in one of the next sessions. And on the other hand, you can use an RDA over time, and that is often done for ecotoxicological experiments. So this technique, RDA, that is done over time is called a principal response curve analysis, and this is very often used for ecotoxicological experiments, and it gives you time-dependent treatment effects relative to a control, and that is often what you want to know in a mesocosm study. Have a look in our course materials. There are extra materials where we described how the PRC is done in R and deliver a little bit more on the background of the PRC. So here we have an overview of the different RDA approaches. On the left-hand side, you see the different approaches. You can use the classical approach, the RDA preserves, where the RDA preserves the Euclidean distance, or you use a canonical correspondence analysis if you rather expect a unimodal gradient. Now you may ask, well, how do I know whether I have a unimodal gradient or a linear gradient? So typically a unimodal gradient would be from a mean or maximum optimum of a species would be 4 standard deviations to each side. So we would check for the standard deviation of the gradient, and that can be done with the so-called detrended correspondence analysis. On the other hand, you could check for higher order terms in RDA. That's discussed in Borchardt et al. We don't discuss this in the course. So if you have shorter gradients, you could use the RDA. If you have very long gradients, you should use the CCA. Another approach is to use transformation-based RDA. That means you transform the data, the species data, for example, before using, for example, the Hellinger transformation. This is often done to bring species data into a linear form. This is discussed in this paper that was provided in the slides, in the notes to the slide. And then we finally have distance-based RDA, where you calculate a distance matrix, and this distance matrix is then used as in the analysis of the RDA after principal coordinate analysis. An example for a study where we employed this is also provided on the slide. There are of course also several other methods that can be used for ordination. We cannot discuss all the different ordination methods in the course. We have no time to do this, but one very widely used method is canonical correspondence analysis. We have discussed it before, and this technique is often used if you assume that you have a unimodal distribution of the species along the gradient of the response of the explanatory variables of the predictors. It's basically an extension of the unconstrained correspondence analysis. The mathematics is similar but not the same as for RDA. It's a little bit more complicated. As I said, we don't go into details here. In R, the model building works in a very similar fashion as that we use for the redundancy analysis and demonstrate in the script. So on the In the notes to this slide, you find a couple of introductions or a couple of texts as introduction to CCA. In case that you don't expect it, you don't have a clear linear gradient or a clear unimodal gradient, then there is also a different technique available. That is constrained additive ordination. That is a technique that is rather data driven, so you fit the responses based on the explanatory variables, based on the predictors, and you don't assume a specific model of the response. So there is no linear or unimodal model assumed. The technique is roughly speaking a mixture of generalized additive models and canonical Gaussian ordination. So canonical Gaussian ordination, you assume a model, a linear response, whether in generalized additive models you smooth a relationship, it's a smoothing technique for data that can be coupled to modeling data via predictors. The technique is computationally quite demanding. This may actually mean that the computer can take up to several hours or even days for the calculation, depending on the data set size and number of variables and species involved and so on. And it's implemented in an extra package in R. It's not implemented in vegan. It's the CAO vegan package. That relates to vectorized generalized additive models. Now that we have discussed these ordination techniques, you may be a little bit confused what to use for which data set and question, and I provide you here an overview that's taken from the book of Legendre and Legendre 2012, and in this overview we'll find, for example, if you want to forecast the structure for what you would do if the response variable is quantitative or if it is qualitative. If your aim is rather to forecast the structure of a multivariate data matrix, what you do. And you will see that several of the techniques and methods that are suggested here we have already used or discussed in the course. For example, if you follow in the upper key forecasting the structure of a single descriptor, then we turn to 2 because we have a response variable as, let's assume it's quantitative. The explanatory variables are quantitative here. Then we end up with multiple linear regression analysis, or if you have high correlations, you use principal component regression that we have discussed, or here that's mentioned ridge regression, but we have rather focused on the lasso in our treatment in the course. And you find different techniques. For example, under 2 and 5 we have logistic regression, that's generalized linear model, and nonlinear regression that pops up here is something that we haven't discussed in the course. That's what you use if you don't have a relationship that you can easily fit from a clear data structure like GLM or linear model. If you have a very different relationship, then nonlinear regression is a method that you should use. And then you have multivariate methods we've already discussed. The linear model for a direct comparison of data via redundancy analysis, and CCA we have briefly touched. You could use tree-like decision models, multivariate regression tree analysis we will later discuss in the course, and ordination methods that we have indirect comparisons, or we don't compare directly data set. Unconstrained methods would be PCA or clustering, and clustering is also a topic that we tackle later on in the course. In the next part, we turn to association measures. An example that we consider here is that we have observed species in 4 streams. They are just called site 1, 2, 3, 4, and we have monitored the abundance of 4 species of stream macroinvertebrates. Now, if you have such a data matrix, you can measure 2 types of associations. On the one hand, you can look at the relationship between objects and between descriptors. Objects would in this case be the sites, and the descriptors would be the species. Typically, you would look at descriptors when you have environmental variables and look at their correlation or something similar. The correlations of species are not that often analyzed. The association measures that you use to measure the relationship between objects are called distance or similarity measures. By contrast, if we look at environmental variables, we are, as mentioned before, more interested at the relationship between descriptors, and we use dependence measures such as the covariance or the correlation between environmental variables. Now let's look at one of the most simple similarity measures, and that is used for presence-absence data. It's called the so-called simple matching coefficient. We have here the case for 2 sites. The formula shows you that you just sum up the presences, so the species are present in site A, in site 1 and site 2, and you sum up the joint absences, so when the species is absent as well in site 1 and site 2, and you sum up the A and D, and then you calculate the fraction to the overall cases. So also considering that of course the species can be absent but in site 1 but present in site 2, and the reverse case. So you should do now the calculation of the simple matching coefficient for the data below, with and without the first and fourth species, and you should check how these species influence the simple matching coefficient. Yeah, the calculation of the simple matching coefficients is obviously quite simple, and if you include all species, you find out that the simple matching coefficient is 0.5. This is because you have 2 shared absences and 2 species that occur only in either of the sites. Now, if you include only the species that occur in either of the sites, the simple matching coefficient becomes 0. Now, ecologically, this means that species absence influence the similarity between sites. And we should halt for a moment and think what this means. This actually means that if we have many species that are occurring in a region, and let's say you only find a small portion of the species pool in the sample, then you always have a high baseline of matching of similarity between sites because you often find shared absences between two sites. That's obviously not desirable. So the joint absence of species does not indicate ecological similarity, and as outlined, as stated before, if you have the joint number of joint absences is largely arbitrary. So which species you have in the species pool depends on the spatial scope of the study. So you could always list, for example, if you investigate insects, you could take all insect species that occur in Rhineland-Palatinate or in Germany on the list. So you have, depending on which scope you have more joint absences in two sites. And that's overall called the double zero problem for these matching coefficients. So widely used similarity measures are discussed now, and one widely used measure for the ecological situation of presence-absence data is the Jacquard coefficient, and as you can see from the equation, this coefficient simply ignores joint absences, what is described in the table as the situation D, where you have the absence for the site 1 and site 2, you have the absence of species that influence the simple matching coefficient, and it's widely used for binary data. Note that these similarity indices that ignore shared absences are asymmetrical, whereas similarity indices that include D are symmetrical. So that means absence and presence are treated in different manner. Both of the Jacquard coefficient and the simple matching coefficient nevertheless range from 0 to 1, and you can easily calculate the dissimilarity, Jacquard dissimilarity, by 1 minus the Jacquard similarity. Now we often have abundance data, not just data on presence or absence of the species, and in this case the Bray-Curtis coefficient is often used. In fact, it shouldn't be named Bray-Curtis coefficient. More appropriate name would be to call it Steinhaus coefficient. See the notes to the slide and the literature reference, the paper in the book of Les Genres. And this coefficient also ranges from 0 to 1 if all abundances are larger than 0, which is typically the case, obviously, in ecological situations. In addition, you see that the minimum of between the values of two sides is taken and divided by the absolute sum. So the overall level of abundance influences this index. And if you have different levels of abundances in which a species occurs in a site, then you should transform the data before to reduce the weight of dominant taxa. So as an example, we calculate the Bray-Curtis coefficient without any data transformation before. And you see in the first case we have a Bray-Curtis similarity of 0.025. If you square root transform the data, the Bray-Curtis similarity increases to almost 0.2, and if we double square root transform the data, then we arrive at the situation of Bray-Curtis similarity of 0.4. So you clearly see that transformation has an effect on the index if we have the situation of just like in the example case where we have one species that occurs in both sides and another species that does not occur in the sides but has a high abundance, that this species would strongly affect the overall similarity, and that's also not desirable. So you could actually argue that if you have half of the species in common in both sides, then you should, and in similar abundances, 5 or 10 in ecological terms is very similar, then you should have a similarity that is also close to 0.5, 0.4 something, and not 0.025. Generally, in extreme cases, you may also have the case that you have no two species that occur in both sides, and in this case, for example, you can have a case where one sample hasn't any species, perhaps because of a chemical spill or something. Then you can add a dummy species to the side, or then actually it will be added to each of the samples, the dummy species that just occurs everywhere, and this helps calculation of this coefficient. In terms of transformation, we should briefly mention that the log transformation, which is often used, slightly increases the weight of rare taxa, whereas the square root or double square root transformation does not. So that just downscales the weight of dominant species. For a further discussion on transformations, see the paper that's listed in the notes to the slide. Now, association measures that meet the triangle inequality criterion are also called distance measures. Now we have to discuss what is actually the triangle criterion, and we have to discuss what we mean by which association measures are relevant here under discussion, and that's actually that holds for the similarity measures as well as for similarity measures. It can be transformed into each other, obviously, and it is these questions of interest because distance measures can be used for geographical representation. For example, that's what you want to do in ordination, and the triangle inequality criterion means if you look at this triangle that the distance between AB and the distance between BC should be, the sum of these distances should be larger than the distance between A to C. So you see here, I mean on the slide, it's obviously an Euclidean representation. You see that this is clearly fulfilled, but for some association measures this criterion is not fulfilled, and then these metrics cannot be used for graphical representation, and or you could geographically represent them, but they would not be meaningful. What you see, the distances that you see in the ordination plot could not be interpreted in a proper way. Several of the measures, similarity measures, can be square root transformed to meet the triangle inequality criterion. So if you use them in raw form, it wouldn't work, but there are transformations to achieve that. Now, one of the most common distance measures is the Euclidean distance that I have already mentioned. That's the typical distance that you measure on a sheet of paper, for example, or in any two-dimensional system where you just measure the direct point distances. And you see the distance as a formula expressed. You know this from school, in the two-dimensional case, for example, the theorem of Pythagoras. And you see that the distance between IJ is the distance x minus E1 J1 plus the distance xi2 minus xj2 squared. And that's simply known what you have done in school. In the multivariate case, we have more dimensions, and we have already encountered the Mahalanobis distance that can be used to evaluate the distance between two multivariate vectors, or between two multivariate points that are represented by two vectors. And these measure takes the overall covariance, takes the covariance and correlation among the descriptors into account, which makes the Mahalanobis distance independent from different scales in the data set. If the covariance matrix for two multivariate vectors would be the identity matrix, so you have the matrix where all you have only ones in the diagonal and zeros in the upper and lower triangle from the diagonal. So that means there's any collinearity, any covariance. They are exactly orthogonal, each of the dimensions. Then the Mahalanobis distance and the Euclidean distance are equal. So the Euclidean distance, or what follows from this, is that the Euclidean distance is sensitive to outliers and depends on the scales of the descriptors. So for species data, the measure is typically dominated by the species with the largest absolute difference in the abundance, unless the magnitude of absolute differences are similar between species. And this is highlighted in the so-called species abundance paradox. You see here a matrix where we have a species site matrix for three species in three sites, and we see that actually a site 2 does not share any species with site x1 and x3, so there is no shared presence of species. However, when you now use the Euclidean distance for calculation, then we see that, you have here the distance matrix between sides, and a smaller value means higher similarity, or smaller value means smaller distance, so sides are more similar. Then you see that side 1 and side 2 have the smallest distance. And that's surprising in an ecological sense. It would mean that the side 1 and side 2 that don't share any species are ecologically more similar than two sides x1 and x3 that have all species occurrences, so presences and absences, in common. So that makes not much sense ecologically. And from this example, you see that the Euclidean distance is problematic for ecological data. We have just discussed a few similarity measures, just two widely used measures for abundance and for binary data, and there are many more association measures that we don't discuss in this course. See Legendre, the 2012 textbook in chapter 7 for an overview of, they give an overview of 20 to 30 different distance measures. Now you might wanna ask, well, how do I select a distance measure or a similarity measure? And this could be done by first checking the literature of your scientific field. They are used in many scientific fields, and you may want to stick to the measures that are commonly used, and as long as they make sense. Don't just repeat something because it has been done in the past. And you find a key in the Legendre chapter as well. And if you follow this key, you see here, this leads you, depending on the type of data, this leads you to potential metrics that can be used. You find also a more thorough discussion of the background of these metrics in this chapter, so it's highly recommended to read that. In R, you have many packages that include functions for calculating association measures. Some of them allow for weighing as well. And I just give a brief overview here of the different packages. And in case you miss one of the association measures, maybe one of the other R packages has that measure included. So far we have only calculated the association measures and looked at the pure values, but as we have encountered at several parts of the course, visualization is often helpful to interpret data. So the first method of visualize the dependency or the distance or similarity is the so-called heat map. And this heat map converts numbers into colors. And this makes the relationship often easier to grasp. So you can see in this heat map, and on the bottom of this page we can see that in red all relationships that are red mean that the distance is 0. So we have a very high similarity, and obviously you have that along the diagonal because the correlation between the side with each other is 1. And the brighter the values become, the higher the distance is between two sides. So the distance, if you look at the matrix with the values, we see that side 3 and 4 have a distance of 0.98, so they have more or less any similarity, and we see that this converts to a white color on the heat map. So heat maps are much easier to interpret than just the numerical column. Another method to evaluate the distance between sides is ordination. We have encountered ordination already, and that works, and that is something that I've already mentioned before, especially if we have a measure that meets the triangle inequality criterion, because otherwise we have no clear geometrical interpretation but of the distances that we see between sides. And here we have the example of a non-metric multidimensional scaling. It's a method that we will further discuss. So non-metric multidimensional scaling is an unconstrained ordination method that is particularly used to visualize distances between the distances for similarity matrix or distance matrix, and it's based on ordered distances. It is the non-metric version of multidimensional scaling. Multidimensional scaling is also called principal coordinate analysis, and principal coordinate analysis preserves the original distances between objects, whereas in the non-metric case that we use here, we only preserve the ordered distances. What means ordered? That means the first and the second distant objects, the highest distance, the first and the second highest distance are also the first and the second distant objects in the geometrical representation, but the relationship between these distances may not be the same. For example, the highest distance between two objects, maybe let's say a distance of 0.9, and between the second distance would be 0.5, so that's almost twofold the distance. So this twofold relationship may not be preserved, but we would see in the graph that nevertheless the distances preserve their absolute ranking, so highest distance and the second highest distance in the ordination diagram. So by this loss of precision, we gain some robustness to outliers, and we can more easily achieve a two-dimensional representation of distances than if we use the metric version. In addition, the distance matrices that we use don't require linear gradient as in the RDA case or PCA case. It just requires monotonicity for the transformation of the distance matrix to a lower dimensional space. And it's overall regarded as one of the most robust unconstrained ordination method and can be used with many distance measures. So it's overall suitable for ecological data. It's based on the distances, and through ordering the distances we lose obviously the metric dimension or the metric aspects, and that means we don't have a method that is based on eigenvalues, and we don't have information on the partitioning of variance. Let's briefly visualize what is the challenge of representing higher dimensional objects in lower dimensions. This is shown here. We have 4 points in a two-dimensional space. These are converted into absolute distances, and we can see that we can approximate these absolute distances in a one-dimensional space. Well, it's only approximated. We can see that if we just look at the order distances, for example, side 2 and 1 and 2 have the largest distance. This is preserved here. Side 3 and 2 have the second largest distances according to distance metric. This is also approximately preserved, although not absolutely, because in this representation, one-dimensional representation, the distance between side 1 and side 4 might be similarly high. Now, in another situation, the metric representation may be much more difficult, and this is shown on the next slide. Here we have these 4 points, all in a relatively similar distance, and this makes it unfeasible to portray this appropriately in a one-dimensional space. So the metric representation does not work out in this case. In addition, the non-metric representation would be relatively difficult. It could be achieved via NMDS to portray this, but the properties of this geometric representation would not match very much the original distances. So the differences would be relatively high. The goodness of fit measure would indicate this. For non-metric representation, that's something that we will discuss later. Overall, we can nevertheless say that NMDS has some more flexibility because it does not preserve the absolute distances between objects but only ranked ordered distances, and that's therefore easier to reduce the dimensionality. Let's quickly look at the steps of the NMDS algorithm. First of all, the distance matrix is determined from the raw data. That's something that we have discussed before. You have to use a distance metric and calculate this matrix. Then you choose an initial configuration in a lower dimensional space. That's often done based on principal coordinate analysis. So you place the different sites in a lower dimensional space and you determine the distance matrix for this initial configuration. Now, this configuration obviously is unlikely to match all the ordered distances, so you determine the so-called disparities using monotone regression. The disparity matrix is the next matrix that is produced in NMDS, and the monotone regression is explained on the next slide in more detail. And after this has been done, then you try to find a new configuration with higher similarity to the initial distance matrix. And after this has been done, you go back to step 3. And if you don't find an improvement on many iterations, the number of iterations can be set for the algorithm, then you move on to 7. And that means you evaluate the goodness of fit of the final configuration that you have found. So let's scrutinize a little bit the individual steps from the distance to the disparity matrix. As we discussed, we start with the raw data and calculate the distance matrix. So the first matrix that we have is the matrix of the raw data. Then you have the distance matrix for this data, that's shown here. And now we look if we look in this matrix, we see that obviously the distances are 0 along the triangle, along the diagonal, and the upper and lower triangle are identical. And we have the distance for, let's say we have site data, then we have the distance between 1 and 2 is 9, between 1 and 3 is 4, and so on. And the ordered distances of the distance matrix are shown below in the center of this slide. So we have the distance 2, 4 is smallest. The distance is just 1. If we look at 2, 5, that's the distance of 2. We look into row 2 and column 5. That's the second lowest. Distances 2 and 3. That's distance 3 is the third lowest, and so on and so on. Now we calculate the distance matrix for the initial configuration, and when we look at these distance matrix, we see that still the condition that for the ordered distances that 2 and 4 have the lowest distance is met, because the distance is 3, that's the lowest in the overall matrix. But we see the second largest is not 2, 5. The second largest would be 4, 5. So we see that the conditions for several of the distances are not fulfilled. What do we do in this case? Well, you would continue with a monotone regression, and that's explained on the next slide. The monotone regression is described on this slide. So we have on the first matrix that we see is the original distance matrix between the objects, and below we see distance matrix for the initial configuration. On the right hand side you would see a figure that describes what is done in the monotone regression. Because we know that the ordered distances should be preserved, and we see here that this equals, so we have on the x-axis we have the initial distances, and we have on the y-axis the distances in the initial configuration matrix. We know that here we have some deviation. Monotone regression means that we should have either we should not have a decrease in the distances if we move along the x-axis or from one point to another. So we should have a monotone increase. So the distances should either remain the same or increase. And we can achieve that by pushing the points to the line that's indicated with the arrow. So the three red points, the points that have been indicated with the red circles, are moved to this line. There we have now 3 times the same distances, and then the monotonicity criterion is fulfilled. We don't have a decrease in distance along the x-axis anymore. And we obtain the so-called disparity matrix, and that is given by the 3 points. Now along this line, the distances for these 3 points are now all 4.7, and the ordered distance criterion is fulfilled. Now this slide shows that again, that in the ordered distances of the ordered distance of the disparity matrix now approximately fulfill the ordered distances of our original data distance matrix, except for the fact that we allow for lower or equal than the other distance. So as we had the ordered distances before, one distance was always lower than the next one. Here we say the distance in the disparity matrix needs to be smaller or equal than the other one. So the distance between 0.2 and 3 in the disparity matrix needs to be smaller than or equal than the distance 1, 3, and so on, and that's fulfilled in the final disparity matrix. Now obviously we could, by averaging the points and working with ordered distances that can be smaller, equal than, we can more or less always find a representation, a non-metrical representation. The question is, how good is this representation? So we need a goodness of fit measure, and this goodness of fit measure expresses the distances in the original data matrix, and so subtracts the distances in the original data matrix with the distances in the disparity matrix and takes the sum of squares. So we are familiar with this way of determining the goodness of fit. The square distances are used, the square differences are used of the distances in the two matrices and divided by the sum of the overall squared distances to scale this by this level of the distances. And this so-called goodness of fit measure is called stress 1. And you see on the right side of the slide, you see values of stress 1 and an interpretation how good the configuration is. So generally, as a rule of thumb, people say, or you find in textbooks, that the value of stress 1 should be lower than 0.1 to yield a good representation of the original data. NMDS is implemented in R in the Wigan package that we have used before. The basic function for NMDS is so-called mono NMDS, but that just runs the NMDS algorithm, whether we have a meta MDS, that's the so-called shotgun method, because it implements a couple of different techniques that are just used, and as a shotgun shoots very broad and not very precise. The method is called like that because it's also a very broad method that just couples a couple of techniques but may not be very precise for an individual data set. But often it does the job and it's very handy to use. You also see here the functions and packages that can be used for metric multidimensional scaling. Since meta MDS is often used, let's quickly discuss what this shotgun method does. First of all, it checks whether the data should be transformed, so either square root or Wisconsin double transformation is applied to the data. Have a look at the help file of meta MDS, how this is decided. And then the distance matrix is calculated based on the selected similarity coefficient, so it also does calculation of the distance matrix. Then you adjust, there's an adjustment if there are no shared occurrences of species. So we had the discussion that we might have a site that has no species that has been found, for example in a spill or something. Then we add a dummy species to the data set. Then it has several random starts to find an initial configuration. It also centers the results and rotates the ordination so that you have the highest dispersion, the highest distances on the first axis, although remember that we cannot interpret the distances in a metrical way. It's not eigenvalue based. We cannot say anything about the relationship between the first and the second axis and absolute variances. Nevertheless, we can see from, if we look at the ordination, we can see that the distances are slightly higher along the first axis than the second axis, but not interpreted in a proportional way. Then the resulting data is scaled. That means that one unit along the axis means a halving of community similarity or a doubling of the distances between two communities if we look at species communities in sites. And finally, there's the calculation of species scores in terms of ecological analysis as weighted averages of sites. So this is described here with a strong focus on ecology, but NMDS is also used in many other fields, and then you'll have the calculation of some scores for objects or descriptors as weighted averages, or the calculation of descriptor scores as weighted averages of objects in general. Yeah, while there is a high flexibility and it's a highly robust method, there are some limitations of NMDS. First of all, the results depend on the initial configuration. That's why we run this several times with a different initial configuration. You will see that we do this in the R script, in the R demonstration part as well. We have obviously a loss of information when we use rank ordered data, and we cannot infer on the absolute distances. And we have no information on the partitioning of variance. In addition, it's difficult to interpret. The results are difficult to interpret if we need more than 2 or 3 dimensions to have an appropriate geometrical representation. And finally, we can, and it is an unconstrained method, but there are for unconstrained methods also possibilities to fit environmental variables to the resulting ordination. In case of ordered distances, it is more difficult to interpret what the correlation of environmental variables with these ordered distances mean, because we have lost the absolute distances between sides. Nevertheless, it's doable, and I show you in the R script how to do this.